Welcome. Today we are going to learn about the global control network and the global regulators. Global control network, it is also called as the global control system. What do you mean by this global control system or the global control network or the global regulatory system? It is the system, okay, global control system. It is the system which regulates the genes comprising more than one regulon. Okay, means what? Under one regulator, there are the number of genes which are going to be regulated, that it is called as an regulon. And this regulon, okay, this number of regulons, they are under the control of the one more system, and that system, it is a global control system. Now, this global control network may include what it is going to include, or which are the components which are going to regulate this global control network. It is the activators, repressors, signal molecules, two component regulatory systems, regulatory RNA, alternative sigma factors. Now, to know more about the global control system, we are going to take some examples. Okay, and these examples, it is from the E. coli. Examples of the global control system which is known into the E. coli. That is the Escherichia coli. Okay. Now, here the first example, it is a aerobic respiration. Now, we all know that the E. coli, it is the facultative anaerobic organism. Okay, so that is what when there is when there is a presence of the oxygen, it is going to go for the aerobic respiration, and when there is a limitation to the oxygen, then it is going for the anaerobic respiration. That is the both of the systems, they are present into the E. coli, and according to the environmental condition, the E. coli, it is going to go for the aerobic respiration or the anaerobic respiration. That is here the signal molecule, it is the presence of the O2. And the regulator which is going to control this, it is the repressor, and it is the ARCA. And this ARCA, it is going to regulate about the 50 genes. Now, going towards the aerobic respiration, okay, that is the ARCA. The anaerobic conditions induces the ARC two-component regulatory system. That is when the E. coli, it has to switch from the aerobic to the anaerobic respiration. At that time, the regulatory system which is activated, it is a ARC, which is a two-component regulatory system, and the components of this ARC, it is a ARCA and a ARCB. Here the ARCA, it is a regulatory protein, and this ARCA, it is going to repress the aerobic respiration by switching of the genes SDH gene, then there is a GLTA gene, then the SODA, and it is going to activate the genes which are required for the anaerobic condition, like the CYD and the BFL. And it is also going to affect the replication, so it is also going to, okay, it is also going to activate the TRAY genes. Okay, so these are some of the examples. Okay, these are some of the examples which are allowing or which are involved under the regulation of the ARC. There are such a number of genes which are controlled by this ARC two-component regulatory system. Thus, here the aerobic respiration in the E. coli, it is a global control network or the global control system or the global regulatory system which is controlling the number of genes. And here I want to tell you that the regulators which are involved into this global control system, okay, that regulators, they will be called as the global regulators. Thus here the ARCA, which is a regulatory protein, this, it is the global regulator because it is involved into the regulation of the number of genes. About how many? About the 50 genes. Thus it will be called as the global regulator. Thus the regulators, okay, the regulators which are involved into the global control systems, that regulators, they will be called as the global regulators. Okay, now going towards the next one, it is a catabolite repression. We all know well about the catabolite repression. It is the it is the system which is allowing, okay, which is allowing the use of the carbon other than the glucose. When there is a depletion of the glucose in the medium, at that time the organism, it is going to switch towards the another carbon sources, and there are the abundant carbon sources which are going to be utilized by the microorganisms. And here this CRP, which is an activator which is involved into this catabolic repression, okay, it is going to control about the 300 genes. Okay, so here we can state that the E. coli, it can use the carbon sources about 300, because under the CRP there are about how many? The 300 genes. Okay, and here the signal molecule, it is a cyclic AMP levels. Thus, this catabolite repression, it is a global control system, and the CRP, it is going to be what? It is going to be a global regulator. Next, there is a heat shock. Okay, in the heat shock, there is the signal, it is a temperature, and here there is a use of the alternative sigma factor like the RPOH, RPOE, and about the 36 genes, they are regulated by the heat shock. Then there is a oxidative stress. Oxidative stress, it is a global control system whose signal, it is oxidizing agent like the hydrogen peroxide, and here it is the activator, and that activator, it is a oxy-R, and about how many? About the 30 genes, it is regulated by this oxy-R. Now here the antioxidant enzymes like the catalases, glutathione reductases, alkyl hydroperoxide reductase, such a type of the enzymes which are involved into the oxidative stress, it is going to triggered by whom? It is going to triggered by the oxy-R, and thus the response to the response to the oxidative agent, oxidizing agent, it is going to be triggered, and the system which is involved here, it is an oxidative stress system, and here the activator, it is a oxy-R. Next, there is a SOS response which is given by the given when there is the DNA damage, and here the repressor, it is a LEX-A. Next one, which is a stringent response, it is a very wide one. Okay, and it is given when there are the starvation conditions and the regulatory signal molecule. Okay, regulatory signal molecule, here it is a guanine tetraphosphate and guanine pentaphosphate. Now here, when there is a start of the stringent response, then there is the inhibition of the tRNA and rRNA synthesis. There is the activation of the biosynthetic operon for the amino acids. There is a activation of the catabolite operons for getting the precursor for the amino acid synthesis. There is a repression of the operons. Okay, there is what? There is a repression of the DNA synthesis, there is a repression of the cell division, and there is a slowdown of the synthesis of the cell envelope components such as the membrane lipids. Now, when there is a stringent response, okay, at that time the guanine tetraphosphate and the pentaphosphate, it is going to activate the biosynthetic operons for that amino acids which are depleted into the cell. And here there will be the start of the catabolism of the proteins for the getting the precursors for the amino acid synthesis. Suppose the lysine, it is depleted. Okay, the lysine, it is in a smaller amount into the cell. At that time, there has to be the synthesis of the lysine. Okay, and for the lysine synthesis, there is a need of what? There is a need of the enzymes. At that time, for the enzyme synthesis, there is a need of the amino acids. So some of the proteins which are not required by the cell, they are broken down, and amino acids, okay, amino acids, they are present into the cell, which are utilized for the synthesis of the enzyme for a particular amino acid. And like suppose there is a high concentration of the tryptophan into the cell, so at that time there is no need of the synthesis of the tryptophan. So what is going to happen? The tryptophan operon, it is going to be switched off because the sufficient amount of the tryptophan, it is present into the cell. And for the DNA synthesis, again, there has to be the continuous synthesis of the DNA polymerases. Okay, and for that continuous synthesis of the DNA polymerases, there is no sufficient amount of the amino acids. Due to that purpose, there is an inhibition of the new rounds of the DNA synthesis, and also cell division. It is going to be limited. Okay, in this way, the stringent response, it is going to control the synthesis of the tRNA, rRNA, catabolism of amino acid, anabolism of the amino acids, and also it is going to affect the DNA synthesis, cell division, and also affecting the synthesis of the cell envelope component. Thus, this stringent response, it is the global regulatory system, global control system, or it is a global control network. Okay, and in that, the signal molecule, guanine tetraphosphate and pentaphosphate, which is involved, it will be called as the global regulator. Okay, I think you may have cleared the idea about the global control system and the global regulators. Thus, the global control systems, they are here we have taken some of the examples only. Here we have learned about the aerobic respiration, catabolite repression, heat shock, oxidative stress, SOS response, stringent response, and which are the global regulators? ARC, CRP, RPOH, RPOE, activator OxyR, repressor LEX-A, and the signal molecule guanine tetraphosphate and guanine pentaphosphate. Okay. I think you may have cleared all the doubts. Still you have a doubt, you may write down into the comment box. If you like my video, please subscribe my channel and also share my videos with your friends so the knowledge it is going to spread. Thank you.